more this is my pleasure to introduce today Heath Pardoe. Heath obtained his PhD in biophysics at the University of WA and carried out postdoctoral training at the Florey Neuroscience Institute in Melbourne. You probably have seen on the flyers that he was assistant professor, but he's actually just been promoted associate professor. So congratulations.
Thank you.
At the New York University School of Medicine, where he's been working since 2012. He uses MRI to investigate the causes and consequences of epilepsy in the human brain, and he's also interested in developing statistical techniques to assist in planning and carrying out large multi-center neuroimaging studies. Title of this presentation today is Clinical and Research Applications of Quantitative Neuroimaging in Epilepsy.
Thanks, Jerome. So thanks for having me along to talk. I'm actually grew up in a town called Boyne up in Western Australia and went to high school in Bunbury, so it's good to come back here and talk. But it's also the first time that I've given a presentation to an infectious diseases and immunology department, so feel free to pipe up if you have any questions if I don't make sense. So what I'm going to talk about is neuroimaging in epilepsy. Just to give you a little bit of background, epilepsy is a common neurological disorder. It affects approximately 1% of the population. And really then the defining feature of epilepsy are seizures. So although a strict definition of epilepsy is continually getting discussed, a good working definition is if you have 2 or more unprovoked seizures over the course of your lifetime, you're said to have epilepsy. And the 2 broad seizure types are focal seizures, which come from a specific part of the brain, or generalized seizures, in which the sort of abnormal electrical activity associated with a seizure comes from all parts of the brain. So my talk today, I'm going to talk specifically about epilepsy neuroimaging for the first third of the talk. Then I'm going to go into a slightly more technical discussion about how I use large neuroimaging data sets for my work to address some issues associated with translating research findings into clinical practice, specifically the effects of how in-scanner motion can affect the sort of measurements that we obtain from MRI scans, and also strategies for harmonizing MRI scans that are acquired from multiple imaging centers. And the reason this is important for translating research MRI findings into clinical practice is that the sort of techniques that I use rely on a large group of healthy controls so that you've got some baseline levels to compare your values to, and generally clinical imaging centers don't have the capability of acquiring these large control data sets, so they've got to be obtained elsewhere. So harmonization is very, although it's relatively technical, hopefully you'll find it interesting. I find it interesting, but it's a really critical step for translating research findings into clinical practice. And finally, I'll talk about age prediction, which is a sort of relatively hot topic in MRI research at the moment, and that's essentially the way that this works is by taking an MRI scan. You can use fairly fancy machine learning techniques to then predict the age of the individual and compare that to their actual chronological age, and there's a lot of evidence to suggest that differences between those 2 are related to health outcomes. So I'll talk about that towards the end of my talk. That's relevant in epilepsy, but it's also being applied to a whole host of other neurological disorders. So to start at the beginning, so the history of sort of MRI and epilepsy are very intimately linked, and the main reason that an MRI scan is so important for someone with epilepsy is it can be very useful for determining their treatment. So most people with epilepsy, approximately 2/3, are well controlled on medication. I mean, that's the first intervention and the primary intervention that's used for someone with epilepsy. But then there's this remaining 1/3 that aren't well controlled when you put them on medication. And for those people, then you need to start to think about alternative forms of treatment, and the standard treatment for those, if it's viable, is surgical resection. So you try and identify which part of their brain is causing them to have seizures, and you take that part of the brain out. And the one sort of specific form of epilepsy, or the one tissue pathology that causes you to have seizures where this has been very successful, is called hippocampal sclerosis. So these are your hippocampi. And what they figured out, so this is sort of was figured out by scientists a generation earlier than me, in sort of the late 80s and early 90s, was that an MRI scan can be used to identify hippocampal sclerosis. And the sclerotic hippocampus in this case is the left, so the left is on the right-hand side of the screen. And the two features of hippocampal sclerosis that you can identify from an MRI scan are increased T2 signal. So this hippocampus here looks brighter than the one on the right. Hopefully you can appreciate that, maybe up here. And the other feature that you see from what's called a T1 weighted MRI scan, so you can see the contrast between these two scans are different, that this hippocampus is smaller than the contralateral hippocampus. And so hippocampal volume loss and increased T2 signal are the two sort of hallmarks of hippocampal sclerosis. Before they had MRI scans, you didn't have any way of you could identify hippocampal sclerosis, but it was a lot more of a dodgy call. So once MRI scans were able to reliably identify hippocampal sclerosis, it really sort of revolutionized epilepsy surgery, because you could go in with a lot more certainty. And these days, if you're unfortunate enough to have epilepsy and you have medically intractable seizures, but it turns out you have hippocampal sclerosis, your chances of being seizure-free after surgery are fairly high. The sort of seizure-free rates are estimated to be 70 to 90%, which is pretty good. And the reason that quantitative neuroimaging, so these days you don't necessarily need to do any sort of fancy measurements on the MRI scan. You can just look at it, a radiologist will look at it, identify it, and that'll be fine. As long as it lines up with all the other sort of clinical assessments. That's one thing to bear in mind. Often I've been in meetings before where someone will clearly have hippocampal sclerosis, but it looks like the seizures might be coming from somewhere else in the brain on the EEG, in which case they're not so keen to take the seizure out. So that does also happen. But a lot of the quantitative neuroimaging methods that have been developed for hippocampal sclerosis patients. And I'm just going to apologise about this figure. You can see that top right histograms are a little bit messed up. So this is just to describe a novel image analysis technique that has been developed by a colleague of mine that we've applied to people with hippocampal sclerosis. You can see, again, in this case, the left hippocampus is clearly atrophic relative to the right, so that's the affected hippocampus here. The way this technique works is by identifying the hippocampal area using a landmark-based technique. So my colleague Babak Ardakani at the Nathan Klein Institute in the United States has developed this technique. It identifies a bunch of landmarks on the MRI scan, so it means you can identify the hippocampal area automatically. Places a region of interest over the hippocampi. So bearing in mind this is all done in 3 dimensions, this is just a representative slice. Once you've identified this hippocampal region of interest, you create a histogram over that area. So I'm not sure how familiar you guys would be with this sort of thing, but essentially a grayscale image like this, all the values that are dark are low intensity, and then say white matter, for example, is a higher intensity. So if you look at the distribution of signal intensities over this hippocampal region, you get a histogram that looks something like this. So this higher peak here would be the little bit of white matter that's contained in this region of interest. Most of it's grey matter, and then a little bit of CSF, which is around these kind of ventricle sort of areas. If you look at the same, if you look at a automatically placed region of interest that's been placed on the sclerotic hippocampus, you can see that you get the characteristic white matter peak and grey matter peak, but there's a much larger CSF region within that region of interest. So the technique works by measuring the histogram, automatically detects the main peak, and then identifies a threshold according to a certain rule, which is below that level, and then it just measures the ratio of the total area of that histogram to the area to the left of the threshold. And the idea is that with hippocampal sclerosis, you can see that there's a slightly larger lateral ventricle, and that leads to a higher ratio of area under here. So what that means is then that people in the ipsilateral mesial temporal lobe, which is the affected temporal lobe, what he terms the hippocampal integrity value is significantly reduced. And so this is a way of objectively identifying hippocampal sclerosis without any human involvement. The really novel thing about this technique is that it's extremely fast. So I don't know if there's anyone in this audience that uses traditional sort of quantitative image analysis methods, for example, like FreeSurfer and other voxel-based morphometry techniques, but they can take a long time. Image processing in this case takes approximately 30 seconds. So you're getting to the point where you can do these kind of automated analysis techniques while someone's still in the magnet on the scanner getting their scan done. And you can have an indication of whether or not they've got an abnormal brain, by the time they come out. And that's another important feature of if research methods are going to be clinically utilised. So that's that work. And this technique's also been, sorry, I should mention this technique's also been applied to other neurological disorders as well. So Babak recently got a paper in JAMA Psychiatry, which is a high-impact psychiatry journal, looking at changes in this hippocampal integrity measure within the first 6 months of a psychotic episode. And it turns out that people that have schizophrenia, their hippocampal integrity rapidly degrades within the first 6 months. So as an aside, the interesting thing about hippocampal sclerosis is that was really the basis for setting up a whole bunch of surgical programs across the world. As the years have passed since the 1980s, though, the number of hippocampal sclerosis cases that have come into these surgical centres has declined, and it turns out now, this is even at the start of when I started out as a postdoc, it would have been considered acceptable to write in a paper that temporal lobe epilepsy is the most common form of epilepsy and hippocampal sclerosis is the most common pathology associated with epilepsy, but that wouldn't be correct anymore. So as it turns out, it's quite a rare disorder, and the number of surgeries, anterior temporal lobectomies, which is when you take out the anterior temporal lobe to get rid of the sclerotic hippocampus, is actually reduced over time, with a concurrent increase in surgeries for tissue abnormalities that are outside the temporal lobe. And so this is an example of what you might see. I'm not sure if there's any radiologists in the audience, but for the rest of us, so there's certain regions of the brain in these two. These are two different cases that are abnormal. Can anyone see the abnormality on this left-hand slide? Can you see this bit of the cortex here? See how it's considerably thicker? So that's a cortical abnormality that's the source of seizures in this patient. This one's maybe a little bit more difficult to see, but you can see in this region here, the cortex is a lot thicker, and also the boundary between the white matter and the cortical grey matter is blurred. So these days, rather than necessarily focusing on the hippocampus as a target brain region that's abnormal, we tend to try and develop techniques and look for abnormalities over the rest of the neocortex. And to do that, the main software package that people use is known as FreeSurfer. And it's a very, so the technique is known as cortical surface modelling. It's an extremely powerful technique. So we use a whole brain T1 weighted MRI scan. which has approximately 1 millimeter voxel resolution. So you get an image that images the brain to a resolution of 1 millimeter. The way the technique works is by identifying the boundary between white matter and grey matter and creating a surface out of that. So that's shown in the yellow, with the yellow line. That surface is then, so you can see a 3D model of that surface is shown here. That surface is then expanded out through the cortical grey matter to the outer grey matter surface, which is shown here. And so once you've generated these two geometric models from the MRI scan, you can then use that to quantitatively assess brain properties. The main thing that people are interested in tends to be cortical thickness, so the thickness of the cortex, which is just the distance between the red line and the yellow line, measured over the whole brain. There are other values that you can look at. So as I showed you that slice of the person with the cortical abnormality on the right, they had blurring between the grey matter and the white matter. You can quantify that as well. And so these two techniques can be quite useful for looking for abnormalities in the neocortex as opposed to subcortical grey matter structures like the hippocampus. And once you have these values quantified, then you can use statistics to look for abnormalities by comparing against a group of healthy controls. So it really takes, it means that we've got objective methods to look for cortical changes in epilepsy. So to sort of describe the work that I do at NYU, and I've been working on this ever since I went over there. This is a study called the Human Epilepsy Project. One of the challenges with the clinical treatment of epilepsy is that someone will come to see the neurologist. They might have had 1 or 2 seizures, so they've got epilepsy. The issue is that there's very few methods available to the neurologist at that point in time to predict what their outcomes are going to be. And so, you know, as the clinicians describe it, they'll start seeing someone, and some people will just deteriorate over the subsequent years before their eyes, and, you know, medication doesn't work. You have higher incidences of anxiety and depression, and then some people get put on the right medications and they're fine. So there's very few methods available to clinicians for most epilepsy patients that can be used to predict their health outcomes in general. So the goal of this study was to use imaging as well as a bunch of other tests, cognitive testings to track medication usage, obtain biological samples to assess DNA and RNA, acquire depression and anxiety instruments, and of course track seizures over the course of the 3 years following diagnosis. The reason that we use a 3-year window is because that's the amount of time it takes to figure out if someone's truly intractable or not, approximately. And the goal is to acquire this data at diagnosis and then in 3 years' time, and to see if there is some way that we can develop markers for health outcomes in these individuals. Bearing in mind, as I mentioned before, that you know that 2/3 of these people, 2/3 of the 600 participants are likely to be well controlled, and 1/3 or approximately 200 of the patients are going to do badly. And from a neuroimaging point of view, this is relatively novel. So I guess the point with MRI scanning is that research is really heavily skewed towards people with really bad epilepsy. So very little is known about what people's brains look like early on in the course of their epilepsy, or for that matter, people that have well-controlled epilepsy that have had it for a long time. That's also something that's been understudied, and that's something that isn't necessarily studied in this project. So the hypotheses for this study, the 3 coloured lines, so the green line, the red dashed line, and the red line are 3 potential scenarios for how an individual's brain structure changes over the first 3 years of their epilepsy. So these 2 dashed lines just represent normal aging, because there is, as I'll discuss later on, there is probably a little bit sort of amplified. You wouldn't expect your brain to change that much over 3 years, but the idea is that potentially the green line, which are people that have well-controlled epilepsy, have neuroanatomical changes that just represent healthy controls. The two potential scenarios for people with poorly controlled epilepsy or medically refractory epilepsy are that their brains look normal at the start at diagnosis, but then progressively get worse, or they have abnormal brains at diagnosis, which then change over the course of their disease duration. The idea is that if this red dashed line turns out to be the case with intractable cases, that if we use more aggressive interventions, potentially get people in for epilepsy surgery earlier, that will improve their outcomes earlier, improve their outcomes in general. So just to give some preliminary data from the study, so we're just at the point now where we're about halfway through the follow-up scan. Most of the baseline scans and assessments have been completed. So for this analysis, myself and Dr. Ruben Kuznecki, who's the main imaging principal investigator on the study, sit down every Friday and review all the MRI scans that come in. So what we've found is that approximately 64% of the brains of people that get enrolled in the study appear normal. This is just based on a visual assessment. 20% have what we call diffuse brain atrophy. So that's brains that appear, the way that he describes it are that the brains appear older than you would expect them to be for their actual age. And this will come back to this. That's sort of relevant for something I'll be talking about further on in the talk. And 15% of the patients have what we would describe as focal abnormalities. And so these include what you would call a funny, very scientific term, a funny looking hippocampus, or volume loss. So that's an N equals 12 out of the 241 subjects. Grey matter heterotopia, so that's where there will just be focal grey matter abnormalities, arachnoid cyst, and other. And so you can actually see that for people with newly diagnosed focal epilepsy, there's very few people that have clearly abnormal brains. Most of the time their brains appear normal. And although I'm not going to really talk about advanced imaging techniques like diffusion-weighted imaging or resting state functional MRI in this talk, the hope is that some of these advanced techniques will be able to identify abnormalities in these patients that have normal appearing brains structurally, or in terms of neuroanatomical imaging. Okay, so the issue, however, so the Human Epilepsy Project that I work on, it's 30 different sites throughout the United States and Australia. There's sites in Melbourne and Sydney that are participating, and a few sites in Europe as well. The issue with doing these studies that if you use different MRI scanners, measurements like cortical thickness or hippocampal volume vary between scanners. And so that's just illustrated on this plot. So this is healthy MRI data acquired from a number of different imaging centres. Subjects are approximately the same age. So these are box plots showing the mean cortical thickness per centre. And you can see there's a significant amount of variability. And that's a real challenge for multi-centre studies. And it's worth bearing in mind that most large neuroimaging studies are multi-centre studies these days. So you really need to overcome this variability to optimise your sensitivity for detecting biological changes in any sort of patient group, not just an epilepsy group. So this is some work I did probably 3 years ago or so. So epilepsy skews fairly young. So a number of studies I've done over the years have been in, kids. Something that's always sort of bugged me is the fact that they don't really hold still in the scanner, and so you can, and an MRI scan is similar to a regular photo in that way, in that if someone's moving during when you're taking the image, it blurs, and you get a poorer quality image. And to be perfectly honest, it blows my mind that it took this long for people to look at this systematically. So I decided to set the way that I first approached this problem. I had a student, Rebecca Kaczarski-Heiss, who was going into med school, and I got her to sit down and visually assess about 2,000 MRI scans to say if they were good quality or not, which she did very well. And then I looked at measured cortical thickness and looked to see if poor quality scans had different cortical thickness values compared to high quality scans. And then discussed it with my colleagues, and they said, well, it would be really good if you had a way of quantifying the quality of the scans that wasn't just based on a person's qualitative take on it. And so I went away and thought about it for a while, and this was sort of the best idea I think I've had in the last 5 years. So for a lot of these data sets that we use, these are publicly available imaging data sets. They acquire an anatomical MRI scan, which is the sort of scan that you can use to measure cortical thickness. But they often, or most of a lot of these studies are actually geared towards acquiring what are called resting state functional imaging scans, which are MRI scans where the person lies in the MRI scanner for 6 minutes or so. An image is acquired every couple of seconds, and they use that to measure changes in blood oxygenation in each voxel in the scan, and they're used to infer functional connectivity between brain regions. And that's a very active area of MRI research, but something that resting state functional scan is very good for is for detecting how much the patient moves, because you're acquiring a scan every couple of seconds. So you can use that to measure motion during that acquisition. So for this study, I used the resting state functional scan to just obtain an estimate of how much the person moved, and then use that in a covariate for a cortical thickness analysis to see if there's a relationship between the amount of motion and the measured thickness of their cortex. So all these data sets, the IIID data sets, that stands for Autism Brain Imaging Data Exchange. So this is an autism study that's made their data publicly available. Similarly, ADHD 200, so that's for attention deficit hyperactivity disorder, and the COBRA study is a schizophrenia study. So the total number of subjects was 2,100. So this is getting towards being a very large kind of neuroimaging study, what I guess some people would call a big data study, although I'm a little bit hesitant to give it that phrase. Sometimes I do. And so to use this data to obtain a motion estimate in each one of these subjects, measure the thickness of the cortex and see if there's any relationship between these two, also taking into account other important variables such as age, gender, scanning site. And so this is what we found. So what's most relevant for multi-center studies, and really quite surprising to me, was that for all these graphs, the amount of motion is shown on the y-axis, and then different relevant variables shown on the x-axis. So in this case, diagnosis. If you feed these into a multilinear regression model, it actually turns out that both the ASD and I believe the schizophrenia groups have increased motion compared to the healthy control group, so meaning that these participants moved more during the scan than healthy controls. Perhaps unsurprisingly, you can see that for kids, they move more during the acquisition than healthy controls, and in my experience, I've got like a kind of heuristic rule that when you're running an MRI scan, if the person's older than you, they're also going to be less compliant, because they just tend to be, in the case of epilepsy patients, they tend to have been just a little bit fed up by the amount of studies they've had done on them. So they tend to move during scans, and that shows up in this graph as well. I didn't run all the scans, but that's just what the curve looks like. Males move more than females, bearing in mind that this was generally the subjects were quite young, so you could probably say young male kids move more than females. But the other really important thing for multi-center studies is there's a significant difference in the amount of motion during a scan across sites. And so that probably just relates to different quality control procedures at individual scanning centers. And that's a really important thing for multi-center studies because so much is just determined by how they run the MRI scan. The issue being that there's no quantitative methods for assessing the quality of an MRI scan. It's really just dependent on the scanner operator to say whether or not they think the scan looks good or not. And that can be quite a difficult call to make, particularly in subtle cases. And then if we look at the relationship between the amount of motion during the scan and the cortical thickness over the whole brain, you can see that nearly all regions of the brain, so the parts that are a hot colour, if someone moves more, then the apparent thickness of their cortex is reduced. There's parts of the motor strip and the occipital lobe where that relationship's reversed, so if they move more, these regions appear to be thicker. And these parts of the brain, the cortex is actually very thin in the first place relative to the other brain. So there's kind of like a reversal of the effect. But the main point is that if people move more during the scan, their cortex appears to be thinner. And most neuroimaging studies in neurological disorders report this finding in their patient groups. So nearly all studies have found that for any disease, the cortex is thinner than their healthy control group. So this is quite an important study because it sort of suggests that motion might be a source of false positives in these studies. And I'm talking about a lot of studies. And you still see studies coming out. I mean, it's only been, I should also point out that since I published this paper, it's been confirmed in about 7 or 8 different independent groups as well by other groups. And it's safe to say that it's generated a fairly strong response from people. Some people get quite upset when you're suggesting that the detects they're affecting are due to motion rather than actually some sort of biological effect. But the issue with this study and with other studies are that you're just inferring the amount of motion during the MRI scan. There's no direct measure. So in my case, it's from a resting state scan that's not done. It's done in the same session as the structural MRI weighted, the structural scan, but it's not acquired at the same time. So I just thought I'd include this slide because this data was just acquired a couple of weeks ago when I was in Melbourne visiting my own lab, my old lab, sorry, not my own lab. This is my brain, so I was the first test subject for this project. My old lab, so that's Graham Japson's laboratory in Melbourne, they've developed a technique. They do EEG inside the MRI scanner, so they do functional imaging while people have got EEG leads on, so that theoretically you can look at functional brain changes during a seizure or during spiking activity. As it turns out, they have a very nice technique for measuring motion in a scanner, and it's sort of like a physics 101 problem. You know, an MRI scanner is a very strong static magnetic field. If you put a coil inside the magnetic field and move the coil, that induces a voltage in the coil. So the way this scan worked was that the techs strapped carbon fibre loops to, I think, just above my ears and the top of my head. And then I lay in the scan, and we did one scan where I didn't move my head, and then I did another scan where I moved my head periodically, and this was every sort of 60 seconds or so. And it worked out really nicely. So it might be a little bit hard to convince you that that scan's a poorer quality than this scan, but it is. But it's a very subtle difference. And if you measure the cortical thickness using the scan on the right, that's reduced relative to the scan on the left. So, so I think this is a really sort of interesting and cool new project where you can measure the amount of motion during an MRI scan. And if we do this in a number of subjects, then you can then use morphometric properties like cortical thickness to estimate backwards the amount of motion during the scan, and in that way develop a quantitative QA tool that can be used to retrospectively correct data sets that haven't had motion directly measured. So I think that will be quite a useful method in the future with a bit of luck. So just to summarise that finding, the finding I've just been talking about, the amount of in-scanner motion varies by site, it varies by diagnosis, so it's generally higher in disease groups, gender and age. And if people move in the scanner, that affects the measured cortical thickness. So that suggests that in-scanner motion is a source of variance and might be a source of false positives for analyses, but it's still not the only thing that varies between scanning sites. So how are we doing? I'll try and get through this a little bit quickly. So there's alternative image quality, there's additional image quality measures. Some are well known and go back to the very invention of it, MRI. So contrast to noise ratio is a way that you can assess the quality of an MRI scan. You can clearly see that this scan on the left is better than the scan on the right. The contrast between the grey matter and the white matter is higher. And so you would say that this scan is a higher quality than that scan on the right. Similarly, signal to noise ratio, which is related to contrast to noise ratio, but just is the overall signal in the image. You can see this scan's brighter than that scan and therefore higher quality. There are more esoteric measures. So something that often varies between scanners is the signal homogeneity across the whole scan. So if you look at white matter, say in the top right part of the image, you might be able to appreciate that that's actually a lower signal intensity than the white matter here on the bottom left. So there's software tools that you can use now to quantify that signal profile change, what they call low frequency signal changes, and you can measure that and quantify that. That's something that you could have two scanners that are the same make and model, a Siemens Prisma 3T scanner, for example, but the homogeneity profile can easily vary between those two scanners. And that will directly affect measurements of cortical thickness, for example, between this region and across scanners. So some more recent work that I've been doing is looking to try and figure out which one of these, and there's a whole bunch of these sort of image quality metrics that you can measure. The software package that I briefly showed up measures 56 of them, which is great, a great amount of information, but we don't know what the most important one is. Again, I think this might be of limited interest, so I did a bunch of analyses to figure out what the most important image quality metrics are, and they turn out to be the signal to noise ratio in the grey matter and the white matter. One thing that was very encouraging about this analysis are that age is has a very large effect on cortical thickness. And that's very encouraging because that means that biological effects rank quite highly in terms of the things that can affect cortical thickness relative to, scanner-dependent effects. So that actually means that that multi-center studies are feasible. If the biological signal in terms of its effect on cortical thickness is high relative to hardware-related effects, it means these studies are useful. All right, so what I'll spend a little bit more time talking about is MRI-based age assessment. So something that they've known for a long time now are that structural properties of your brain, like, for example, the volumes of brain structures like your hippocampus and your overall brain volume and your cortical thickness are age-dependent. And similarly, I'm not sure if you guys do functional imaging, but for example, functional connectivity between brain regions, they're also age dependent. And so then people have, over the last sort of 5 to 10 years, done these studies that have used an MRI scan. So you take a whole bunch of MRI scans in healthy controls, measure brain properties such as structural measures or functional measures, plug those into a machine learning algorithm and use them to develop a predictive model that can use an MRI scan to predict the age of an individual. So once you've built your predictive model, you can then input patient data to that. And what a lot of studies have found, in Alzheimer's disease, traumatic brain injury, HIV, these individuals have brains that appear older than their chronological age. So there's a bigger difference between their predicted age from the MRI scan and their chronological age. And in fact, a recent study showed that people that have an increased brain age are also have higher risk of early mortality. So this should all be something that sort of, I think, gets missed a little bit in these kind of analyses are the fact that your chronological age is a very strong predictor for disease risk in just about all diseases. So the premise of these studies, you know, neuroimaging-based age prediction is only really going to be useful if it's more informative than chronological age, because chronological age is already informative in terms of disease risk. The reason that, just to show you the sort of mechanism behind particularly these structural age prediction methods, are the fact that, as I mentioned previously, the thickness of your cortex changes as you age. Again, this is one of the interesting things about working in this field. About 5 years ago, if I presented a slide like this, it wouldn't be from this paper because this was published since then. People used to think that your cortex actually got thicker in this sort of 5 to 10 range. So all the plots from earlier papers show an increase up to here and then a reduction. We haven't shown it, but this sort of ties into my motion work before. You know, I was just explaining that people that move more in the scanner have thinner cortices, so that might explain why they used to think that, but now the prevailing thinking is that there's just a general decrease in cortical thickness as you get older. And so we've done a similar analysis in epilepsy cases. So these are histograms showing, plotting the distribution of the difference between chronological age and predicted age, sorry, predicted age minus chronological age. So as you would hope, in an independent group of healthy controls, so this wasn't the same controls that was used to train the model. On average, the difference between your predicted age and chronological age is 0. For people with newly diagnosed epilepsy, so these are the patients that come from the Human Epilepsy Project study that I've been working on, you can see there's a small increase to the right, but that's not statistically significant. And in people with medically refractory epilepsy, so these are people that come to our epilepsy centre for pre-surgical planning, so they've got very bad epilepsy, had it for a long time. You can see that their brains appear to be approximately 5 years older than you would expect, and that is a statistically significant difference. The sort of outcome of this study is that people with medically refractory epilepsy, long-term epilepsy, have brains that are prematurely aged. This one here. I can't remember. I think the N is only about 42 subjects, so it's a little bit hard to interpret that too much. What we're really interested in, we know that that 1/3 of these people are going to be going to eventually go on to this medically refractory group. So what we would be interested in doing is subdividing this group up once we know what their outcomes are like, and seeing if they're people that fall on this right-hand side of the histogram. But that's probably just noise, those bumps. So, you know, people are generally interested in using these kind of predictive models to, you know, see if there's some clinical utility. I think the thing that's worth bearing in mind is that there's still actually quite a wide spread in people that are healthy controls. And so that's probably some inherent variability across subjects, but it's probably also a limitation of the technique as well. So just meaning that your age prediction model isn't super accurate and there's a fair bit of noise. So some work that I've been doing that hopes to kind of try and address this in a roundabout way is to not just develop my own age prediction model, because it doesn't perform, it performs about in line with all the other models that are available. So just to give you an outline of how you train the model. So in this case, I took 2,300 subjects from publicly available neuroimaging data sets, processed them using free surface, so measured their cortical thickness and the volume of subcortical structures. Use relevance vector machines, which is a machine learning technique to predict a continuous variable, so age. For those of you familiar with machine learning techniques, they're often just used for classification, but there are techniques that you can use to predict a continuous variable. And then to develop the model, and then make the model available on a website, on a cloud-based server, running a software package called OpenCPU. So you can go to this website if you're interested, and if anyone's interested in this technique, I can show you how it works. But the main point is that the age prediction model is publicly available. So people, if you've got your own MRI scans here, they do need to be processed using FreeSurfer, which is a little bit of a computational burden, although not too bad. You can then use your own data to predict the age and therefore validate the model. And so I think, moving forward, a lot of this kind of approach is going to be used more and more. Just simply making your, I'm sure you guys are familiar, even in your own research, that there's a big push towards making your research data available rather than just publishing a paper and having people believe your findings. In this kind of neuroimaging predictive modeling field, I think, it's particularly critically important to make your predictive models available so people can validate them externally, because it's a known problem that the models can work pretty well on your own data sets, but often don't translate well to other data sets. That's probably going to be of limited interest. This is just a summary of the model performance with age, so how well the predictions work with age. And you can see that there's a bit of a bias. So for the younger subjects, it over-predicts the age, and for older subjects, it under-predicts. So this is quite a sort of, the idea of developing predictive models from imaging data or from using imaging data in combination with other data is, a very hot topic in neuroimaging at the moment. Depending on how closely you've been following it, so for example, for the HEP study, we're interested in predicting health outcomes in epilepsy subjects. So the question really becomes why do you use this intermediate prediction of age to then predict outcomes in epilepsy? So you could just as easily do this sort of alternative left-handed approach where you just directly use the MRI scan to predict the clinical outcome without doing this intermediate step. This paper, I've got nothing to do with this paper, but it's if you're interested in this work, I recommend reading it because it's a very nice sort of summary of neuroimaging predictive modeling. The way that they conceptualize it is that you're using it to make this intermediate prediction that can then relate to a whole range of clinical outcomes, not just in terms of epilepsy. And that tends to match up with what happens biologically, because what you find is that there's a lot of comorbidities in epilepsy. People with epilepsy often have a whole range of health problems, some of which may actually cause the seizures in the first place. So the idea is that age prediction is an index of more general brain health rather than a specific disease. And I think that's my last slide. Yep. Thanks very much.
Thank you very much for this very insightful presentation.